In this video, we will be going over how to do the Caspase assay. So I will be doing this video showing you the basic steps, and please be reviewing your protocol as well. So I will be moving this video into the hood so you can see what's going on. To begin with our assay, we first need to take our cover slips that we had put into our 6-well dish, and we actually need to transfer them over to our 24-well dish. This will allow us to treat the cells that are on the cover slip with the reagents for the Caspase assay, and to do it in a much smaller volume. So to begin, I'm actually going to load up my 24-well dish with media. So I'm just going to go like this. And I've already have them labeled 1 through 10. And I'm putting 250 microliters into each well. Okay, so I have loaded up all the wells with the media. And now I'm ready to do my transfer. I might go ahead, so I know that I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So I'm going to go ahead and move my dish cover to the side. And I'm going to grab my needle and the tweezers that we had used to fill up our dish previously. And I just realized I really don't need numbers 1 and 2, so I might just skip those 2 and start with number 3. So, but I might not actually need my needle. Let's see here. So we want to very carefully scoot our cover slip over to the side. We could probably just use our tweezers because it's floating a little bit in the media. You're just going to have to use some patience here. It doesn't want to come up. Let me switch my tweezers. We've got a couple different kinds in here. If the tweezer you're working is giving you too much trouble, just come in here and grab another one. One doesn't want to come with me either. So I'm going to grab these with the little hooks and see if that works better. Much better. There we go. Should have started there. So we've all learned a lesson. We're going to use the curved tweezers here. So I have my letter R. I know my cells are on this side. They're not on this side. And I'm going to place this so the letter R is still facing up into my 24-well dish. That's number 3. Okay, I want to keep the numbers the same. In between these, it's okay if I kind of use the same tweezers because we're kind of at the end stage over here. I'm just going to keep working my way through. So when I'm doing this, oh, that's so much easier. 1, 2, 3, 4. When I'm doing this, I always want to make sure the letter R remains facing the same direction. If it gets flipped, I want to flip it back the way it was. As I'm collecting the cover slips, I want to be sure not to disturb the cells that are in there too much. 1, 2, so 3, 4, 5, 6. So I'm already done with this one. I'm going to return these back over here. I'll sterilize that and put it back into that dish in a minute. Let's do this switch around. Okay, we're going to do 7, 8, 9, and 10. So just remember, there's still cells at the bottom of this dish that we're going to be collecting for RNA. So we don't want to be disturbing them too much. Likewise, we don't want to disturb the cells on the cover slip too much. So I'm trying to just grab on a corner and not put my tweezers right in the center of it, but just put it on the corner. It's okay if a few cells get taken off, but you definitely don't want all the cells to come off. There we go. This is going a lot better now that we've switched our tweezers. There we go. That one stood up on the side, so I'm going to push it down. I want to make sure all of these are sitting at the bottom, not sideways or anything, because we will be adding reagent on top of here. I'm going to return these back to the ethanol and put the lids back on here. Okay, so just keep in mind that this dish has cells in it that we are going to be collecting for RNA. So I'm actually just going to set them to the side for a minute while I treat these with the Caspase reagent. And this has an incubation time of an hour, so I want to go ahead and get that going. And then while this is incubating, I can then be dealing with this plate. I can use this plate to take images of the cells so that I have those for my lab notebook, and also I could use those for my lab report or my poster if I'd like to. And then finally we will be collecting the RNA from these dishes. So I'm just going to set this to the side, and then I will be ready to deal with this dish first. To begin our Caspase assay, we will be treating our cells with the flicker reagent. So the flicker reagent will be prepared for you by your instructor beforehand, and it contains a reagent that has the caspase inhibitor sequence, DEVD, which is linked to a green fluorescent probe. So this reagent does have fluorescence to it, and it is pretty expensive. So when we use it, we try to use it in a small volume, which is why we're using the 24-well plate. This reagent is going to need an hour incubation time. It allows it enough time to enter into the cells. It can go through the plasma membrane. And once it does go through the plasma membrane, if a cell is going through apoptosis and it has activated caspase pathways, the FLICA will actually bind to an active caspase. So to do this process, we have our FLICA reagent, which will be given to you by your instructor. I have my light turned on here in this video, but typically I would do this assay with the lights off in the hood. So just keep that in mind. So the very first thing we need to do, we have RPMI in here with our cover slips. So we actually want to take the RPMI out. And so we can do this just by using our pipette and just kind of going one by one. I can select, I had this on 250. I'm going to go ahead and move this up to 1 mil just so I could grab more volume and it can take a little bit less time. So you just hold it at an angle and grab multiple wells at a time. Just be careful not to put your tip in the middle of the cover slip. You always want to be touching the very corner so you're not disturbing your cells. So we wanted to make sure we had media in our well because we don't want our cells to dry out. We want to keep the cells alive and happy. And it gives us a chance to kind of wash them off. Okay, so we're finished with that. Our next step is to simply just add the flicker reagents to the cells. So for our assay, we're going to be adding 200 microliters. I'm actually going to, you could use the big 1000 microliter pipette or the 200 microliter pipette. I'm going to go ahead and use the 200 microliter pipette. So I have it dialed in 200, just because it's going to give a more accurate volume. And the 1000 microliter pipette, sometimes when you release your thumb, just liquid just comes up too fast. And I definitely don't want to waste any of this reagent. So pretty much what has been given to you is almost exactly what you need So do number 3 through 10, and just when you release the volume, just release it so it's sliding down the edge of the well. And don't just shoot it directly onto the top of the cover slips, because it could cause some of the cells to fall off. Okay, so that's it. We have already treated our cells with the reagents. I do want to shake it just slightly and observe and make sure that all of the cover slips do have media on top of them. Sometimes there's like a little air pocket or a dry spot in the middle of the cover slip, and the media has a hard time going across it. So make sure that you're doing that. And then check with your instructor. We have foil in the lab, so we would cover this with foil. And then I'm going to put this into the 37-degree incubator for 1 hour. So it has enough time for that flicker reagent to get into the cells and to attach to the caspases. In the meantime, I will be working with my 6-well dish. So we're going to take pictures, and then we're also going to collect the RNA. Okay, so my Caspase assay is in the incubator right now, and I have a whole hour to kill, so I can use it spending time with these guys. So I'm not going to take a video of it, but I would encourage you to first take your 6-well dish over to the microscope, and using the app on your phone, collect images from each one of these wells, and just do it in a way that you are keeping track of which image is for what well. So maybe just write down on a notepad what exactly you're taking, and then just go ahead and organize them as soon as you can after you're done in lab, so you don't kind of forget what image belongs to what. Once you have finished that, now you're ready to collect the RNA. So I've already prepared my tubes, but you would be coming and collecting or preparing your tubes like this as well. And keep in mind, none of this needs to be done in the hood. I'm just kind of set up in the hood right now, so I'm just going to continue to stay here. But just get the 1.5 mil tubes. Use a marker to label them. And you can kind of do it however you want, but I kind of keep it simple on the lid. I just have a number 1 here. I put group 1 so that you could put a group 1 on the tab on all of these, so that when they're in the freezer box, it'll be easy to identify which ones belong to you, because everybody is going to have a number 1 through 10. On the side, you can give it some more detailed information. So a lot of times I repeat what I have on the top in case it comes off. But I have group 1, number 1, and I put what is number 1. It's the control. What did I collect? RNA. I probably should have put RNA lysate, but I think for this class we will know what it is. And then I usually put the date, so 9/22/20. So I do usually label my tubes like this. As long as you have your group number and the actual sample number, you will be good to go. Sometimes it is good to include the whole label, because if by chance part of the label starts to come off, then you have enough to kind of figure out what sample that is. So make sure you're doing that for all 10 of your tubes. Make sure you label clearly and have your group number on there. Okay, so your next step is to remove, I forgot to get a pipette. You want to remove the media that is on here, and then we will be adding in the RNA lysis buffer. So you can do this in a quick way, because we don't need to actually be sterile or anything here. So I'm actually just going to collect like this. I'm just going to go from well to well to well, and I hold it at an angle, and I really don't want any extra liquid to be left over, because that does tend to mess this up a little bit, because it won't fit in the tube. So I can just do that, use the same pipette to go from one well to the next. I really have to watch my pipette. See these bubbles coming up? The bubbles are fine, just don't let the bubbles get up past this red line, because then it'll gunk up our filter and it won't work anymore. Okay, so we do want to put this in the biohazard trash. And now we're ready to add our RNA lysis buffer. So we are going to add 300 microliters. Just double checking that that's the right amount. So I'm going to just dial this down to 300. So 030, see if you can see that. And that'll be 300 microliters that are going to go into each one of these wells. So I can use my big pipette. And again, I can just go from well to well to well. Just scoot things over so you can see. And I don't have to be keeping anything sterile at this point, which is nice. I can kind of relax a little bit with that. So I don't know if you guys can see, but I'm just going to go. I'll probably have you work out of a 15 mil tube. But we're just going to go down the line 3, 3, 3, all the way down. I do not want to touch the tip to the bottom here because I don't want to contaminate my bottle. As long as I maintain a safe distance, I'll kind of be okay. Alright. Boom. That's all done. Okay, so now I've added my RNA lysis buffer. I usually give this a good 3 to 5 minutes. Technically, it doesn't even need that long. But I do just make sure all of the cells are covered by shaking it like this. And I have to watch it. Sometimes there are those dry spots sometimes that don't get covered. So I will set that down here, and I'll just wait about 3 minutes. I'm going to ask you guys to take this over to the microscope, and it would be great if you could take another image so you can compare your before and after image, before the RNA lysis buffer and after, and it's pretty amazing the difference it makes. So the RNA lysis buffer will lyse the cells and release all the components of the cells into the buffer, okay? And so you can just kind of keep shaking it like this a little bit. I'm going to push pause and come back, and we will do the collection. Okay, so we are ready to collect our RNA lysate and put it into our tubes. So I have 300 microliters in here. I usually set this just a little bit higher, just to make sure I get everything, but not too high because that kind of helps introduce bubbles. So I will get my tip. All right, for sample number 1, so if you guys can see that, I can pipette up the liquid. It's in my tip. I'm going to wash the well, so I can make sure all those cells and cell lysate are coming with me, but I want to be careful not to make too many bubbles. If I pipette too aggressively and introduce too much air, I get a lot of bubbles. And then I'm going to put that into tube number 1, and I will discard my tip. And go ahead and close it so you don't accidentally add the next well to that tube, which is easy to do once you get going and if you have a lot of samples. Okay, so I will keep going with this. You don't need to watch the video of me doing all of the tubes, but you would just keep continuing until you got to number 10. Once you get to the end, we will be putting these in the freezer in the -80 and storing them. They can stay in the -80 for quite some time before you finish doing the RNA purification process. So it's a great way to store your cell lysate so that you can kind of proceed on another day. So be sure to just give these to your instructor when you're done, and they will be put in the -80, and we will come back to class and finish that on another day. Okay, so now we're going back to our caspase assay. Our hour is up. We can remove our cells from the foil. And so we will just place that here. We'll keep our foil to the side because we're going to reuse that. We want to remove our Flicka reagent. And then we will be adding a Propidium iodide reagent. So, sorry, I bumped the camera. Again, we will just go from well to well, removing our media. We're going to use 250 microliters, so on the big pipette it's 0 to 5. And those tips, sometimes you have to just push down a couple times to make them go in correctly. You might want to just mix it a little bit before you start. And again, we can just go from well to well. No need to change our tip or anything. And again, we want to, I should have been tilting this to the side. I was trying to hold my arm so you could see it, but let's tilt it to the side, and that would be preferred, to have you go down the side of the well and not be blasting this solution on top of your cells. You just want to do everything very gently. You don't want to disturb the cells. And again, we want to just mix it and make sure that our cover slips are covered with the media. We're always trying to save reagents, so that's why we don't use an excess, so we're just barely at the point where we can cover everything. So we'll make sure that that is covered. And we'll return it again to our foil. Let's put that in here. Always remember where the top is so you don't accidentally hold it upside down. And this one will be incubated for 2 to 10 minutes at 37 degrees. And then we will continue with this assay once that incubation time is up. Okay, our incubation time is up for the Propidium iodide, and it's time to wash and fix the cells. Okay. So to begin, I'm going to remove all of the media that's on the cells, the staining solution. So I've discarded my PI staining solution, and now I'm going to wash the cells. So I'm going to use 250 microliters of my wash solution to just wash the cells and prepare them for fixing. So again, I'm going to do the same thing, going down the side of the cells. Side of the wells, I mean. I will gently rock them back and forth. And I'm going to just go through and remove it. And that was one wash. So now I'm going to do another wash. If you have to set it down because you can't hold it all, that's fine. Just put your tip up to the side and let it wash down the side of the well. And that'll be fine too. Alright, so that's my second wash. I'm just going to remove the wash solution. Some of these, there's a little bit left over. I can always go back and grab the rest. Okay. This next step is the fixing step. So when you add the fixing solution, it has paraformaldehyde in it. And the paraformaldehyde really just like locks things into place, and it kind of freezes the cells in time and freezes proteins and things like that. So we really got to be careful with this reagent. It's pretty toxic, so you definitely want to be using, I would recommend you to use a chemical fume hood, because it's going to protect you from the fumes actually more than a tissue culture hood would. I'm just going to keep this video going here. So we have our fixative paraformaldehyde, and keep in mind I will also be asking you to keep the lights off in the hood when you're doing this, because we are working with fluorescent reagents. So this one is actually 500 microliters, and I'm just going to add it to my wells. It. I just want to make sure that they are covered. Okay, we have another incubation step here. I'll just put my caps back on. We do want to keep this in foil, protected from the light. Ever since we added that flicker reagent, we've got to really just keep it protected from the light. So I'm just going to keep using that same piece of foil over and over again. It is going to sit here for 15 minutes. And during that 15-minute time, I can be labeling my microscope slides. So I'm ready to go for the very last step. So I will pause this video and come back. Okay, we're going to continue on with this after the fixative step. This is nice because at this point our cells are fixed. You can kind of relax and breathe, because pretty much you can do almost anything and they will be fine from this point on. So that's kind of nice. So I will be providing you with a weigh speaker for this formaldehyde. So definitely ask where we should be discarding the formaldehyde. We're going to go through the same process as before, removing. Making sure our pipette tip is not touching our cover slip. If a little bit stays in there, that's fine, because we're going to do a couple wash steps. We are going to wash with 3% BSA. So let me just get back here. So same as before. We are adding BSA to each well. We're just going to shake it and wash it. And I'm just simply going to discard of the BSA. Using the same tip makes it go by a lot faster. And you can just go like that. And just repeat that one more time. Oops. Getting a little overzealous over here. I do realize this is not the most exciting video to watch, so if you've made it to this point, you deserve an A. Okay, that was our last wash for the BSA. Shake it back and forth. Just going to remove it. It's just really going to keep that, just wash that formaldehyde right off. We really don't want to have any traces of that around as we're putting it on our microscope slide. We are going to just add 250 microliters more, just enough to keep everything kind of wet, submerged. Okay, that's it. I made it. Alright, so I have my BSA in here, my cover slips are still there. And now I'm ready to put my cover slips on the slides. Alright, here comes the grand finale for our experiment. We are going to be mounting our cover slips onto slides using this Propidium Gold Anti-Fade. It has DAPI reagent in it, which is going to stain the DNA in our nuclei blue, which we will see when we do our imaging. And here's our microscope slide here. So you can just, during one of your incubation times, be labeling your slides. You can write on the blue part. Definitely include your group number. You're going to be putting 2 cover slips on here. So put 2 labels. The one on the top is the first one. The one below it is the one that's going to be lower. So I put 1 cells, 2 lipo. Of course, I made a mistake. Almost didn't spell cells correctly. I was putting control. I realized I needed to put cells. Okay, so we're going to take our pipette. I have it set to 5. So this is a 1 to 10 microliter or 2 to 20 microliter pipette. And then I will probably be giving you a 1.5 ml tube with this reagent in it. You can see it's real globby. Right? It has some, I believe it has glycerol in it. And glycerol can be challenging to pipette sometimes. So I'm going to just put this on the cover slip, or on the slide where I'm going to be putting my cover slip. And I'm going to need 2 of those separated apart. Okay. Now I need to go collect my cover slips. It should be interesting. It's a little bit challenging because we've got this teeny tiny little dish here. Okay. Oh my gosh, I almost got lucky. It almost stood up on its edge. And again, just really be careful. I just kind of get it to stand up, and then I grab the edge here. I should have had a paper towel with me so I could just tap the edge of this. Let me see if I can grab one with my other hand. There we go. Okay, so I'm just going to tap this very slightly on the edge on this paper towel, remove some of the extra media. Okay. So look, I have the letter R to where I can read it. So this is the side that my cells are on. They are not on the opposite side. So I want my cells to face down. I want them to be in the middle. So I'm going to take note of which side the cover slip I'm dealing with here. I'm going to hold it so that one edge is kind of in that little dot of DAPI fixing reagent there. And I'm going to just slowly lower it like so. And I see a little bubble here, so I will use my tweezers to just tap and see that bubble popped right out of there. I don't know if you guys can see it on the video, but I have like a little bubble. It was here, but now I pushed it out. So I don't want to go too crazy with this. A couple bubbles here or there, it's going to be fine. But if there is a big bubble I can get rid of, then I will do that. Okay, so I will do one more for this video. Hopefully we'll have some good luck. There, got it to stand up. I have my letter R facing me so I can read it. My paper towels come flying around in here. I'm just gently going to tap. If you forget to do that, that's fine. There's not so much liquid here, it's going to be a problem. And then I'm just going to slowly lower it again. Give it a couple taps. Remove those air bubbles. Alright. Okay, so the very last step, you're going to do that for all 10 of them. You will be putting them into one of these containers for them to dry. So lift them up, place it inside of your container like so, close the lid, and give them to your instructors so that they can dry overnight. We can actually wait till next week or even another week from then to image these on the confocal. So they'll be in here. It takes about 24 hours for them to completely harden, where they're safe, where the cover slip definitely will not fall off. And we've got it arranged so that now our R is upside down. We can't read it correctly, right? Because we've flipped it over so that our cells would be facing towards the microscope slide. We definitely do not want the cells to be here on the surface facing the outside world. When it comes time to imaging, we're going to be spraying the slide with ethanol, wiping it with a chem wipe, and that could certainly remove our cells. So you just need to do that for the remaining of your samples, and then you are done. If you have any questions, please let me know, and thank you for watching.